Good evening, ladies and gentlemen. Welcome to the Melbourne Brain Centre and to this ICT for Life Sciences Forum presentation. My name's Luan Ismail, I'm the forum convener. The ICT for Life Sciences Forum is a sponsor-funded initiative to bring together the community of interest in Melbourne around the convergence of the life sciences and the physical sciences, engineering, computing, mathematics, physics. The forum was established in 2008 and we have events like these on a very regular basis to bring you thought leaders, present them to you to describe some of the quite amazing things that are happening in this area of convergence. This evening we have Graham Cameron, who's from the UK and was really the founder of the European Bioinformatics Institute. Over 20 years experience in that field, and he will give us an overview of some of the global infrastructure in this space and how Australia might play a role. Please welcome Graham.
Thanks very much. I'm delighted to be here and have the opportunity to talk about this. I'm going to talk about bioinformatics. This involves biology and it involves informatics, and usually I'm talking either to biologists or informaticians. When I'm talking to biologists, I pretend I know something about informatics, and when I'm talking to informaticians, I pretend I know something about biology. So this forum may be a little challenging for me in terms of the audience, because I gather I've got both here. Bioinformatics is central to modern life science. Basically, what we mean, it's kind of been hijacked by the biomolecular sciences to refer to the methods and the data and the tools to exploit the data that is done in modern high throughput science. It actually requires a fairly extensive information infrastructure to enable quite a diverse community to take advantage of what are now a wide range of heterogeneous data collections. And it's about this infrastructure that I'm going to talk primarily, and I'm going to be arrogant enough to make some suggestions about how Australia might be involved. And this may well be a case of fools rushing in, Scottish fools rushing in where Australian angels fear to tread. But first of all, I want to sort of convince you that this matters. The starting point for this was the DNA databases, and DNA is rather beautiful. This is it binding to p53. At least I think that's a rather beautiful representation of it. But the geeks can make a pretty good job of making it boring by turning it into a list of A's, C's, T's, and G's. From the DNA, of course, all the biologists know you then have to transcribe to RNA in the typical situation, and then and makes proteins. So it's crucial to living systems, and the protein people manage to be slightly less boring with their sequences in that they extend the alphabet to 20 characters. But again, these molecules, when they form three-dimensional structures, are rather beautiful. And these molecules interact with each other and perhaps with small molecules. This is a drug, an AIDS drug. And these reactions form a set of pathways and chains of things that happen, and they are basically the entire choreography of what goes on in cells. And I'm sure the biologists could explain that far better than me, but that's the space in which we're working. My background is in the EBI, which is a major part of the global information infrastructure, and the way we think about that, our task, we used the phrase the central dogma, which was something that Jim Watson, I think, first said, and that kind of goes from that genomes contain genes which are transcribed and the transcripts translated to protein sequences. These form three-dimensional structures. They interact with each other with small molecules, they form pathways, and these combine to build systems. We're in the situation where today's science has in fact enabled us to assemble massive information collections at all parts of that cascade. And in fact, if you were at the EBI, you'd see this slide that says that we've got genome databases, genomics, we've got pathways, we've got models, chemical entities of interest to biology, and so on. So all of this information, which is crucial to biology. And if you looked at the tools that they build at the EBI, you'd find that actually they're organized in a way, if you do searches, that reflect this central dogma. Now, this is the basis of all living systems. I mean, people will tell you that there's differences, but seen from a bit of a distance, they're minor kinks in terms of the systems. And exploiting the understanding of such systems has an enormous and an increasing societal benefit or impact, and the obvious application areas are in human health and well-being, in medicine and the biology, so human biology and the biology of pathogens and design of drugs and so on. But in fact, the information has implications for our relationship with the entire biosphere. And if you look at these databases, you'll see everything from crop plants to rainforests, from coral reefs to brewer's yeast, farmed fish and fruit trees. So it's kind of important in its application areas. And it's all basically about the molecules, and for the human or for any other organism, different molecules are required in different tissues at different developmental stages in response to different environmental factors. And so there are differing levels of transcription and translation, and these affect what goes on in living systems. And there can be genetic differences which cause variation, some of which is benign, some of which causes pathologies, and some of which can influence the propensity to pathologies. So by analysing genomes, genes and gene products, we can understand the mechanisms of health and disease, we can diagnose disease, we can design drugs to treat disease. Now this is all made a lot simpler because biology tends to be all kind of joined up. So molecules that are similar to each other have similar behaviour, they're typically related in evolution, and therefore the understanding of new data in the methodology we use is utterly dependent on comparing them with existing data. So modern research can't advance without access to these data collections for this kind of comparison work. And actually, this is a slide that I started using about 10 years ago, but it still sort of works, is that basically, if today you've found a gene that is important in some distinction, whether it differs in a healthy individual or from a diseased individual, or whether it differs in a high-yield crop from a low-yield crop, or maybe a disease-prone farmed salmon and a disease-resistant farmed salmon, or a crop that will tolerate salt and one that won't, the kind of methodology you go through is the same whether you're dealing with human beings or you're dealing with wheat. You go and look in the databases and try and find similar genes and their products and their functions and their interactions and expression patterns and the processes in which they're involved. And if you can do that, you may get some insight into how to intervene so that you can actually do something to influence perhaps pathways or use drugs to enable you to move, to take the disease from the disease state to the healthy state, or the low yield to the high yield, or whatever. And this level of understanding then enables you to talk about ways that you would influence the organism in question. And this can be, people often think when you say this that you're talking about genetic engineering. Genetic engineering is one of the things that you might do, but the understanding, so it's a rather simple concept. But it's already extremely relevant in human health and well-being. I'm afraid the last time I was talking about this, I was in Europe, but basically, for example, 1 in 10 women in Europe, in the 27 European states, will develop breast cancer before the age of 80. And we can actually now identify genes that cause a propensity to breast cancer. We can analyse tumours, work out what's gone wrong, that can influence the kind of treatments that we'd adopt. So it's important, things like that. It's also important to tracking the source of infection. So if you take the RMSA, we can look at different strains, we can look at different variation, and this is a problem in hospitals throughout the world. And we're at the stage now where we can work out exactly from which bed to which bed in which hospital this has been transmitted. This gives us an understanding of how the disease is spreading and enables us to build better measures to stop it happening. And this is something that is relatively easy nowadays if you have the relevant databases to do it. If you look at crop plants, we can look at which variants are likely to withstand particular climates. We can identify drought-tolerant variants, and we can do more, as I indicated earlier, more efficient crop breeding. Another interesting application of this is simply in identifying organisms. So for the Barcode of Life project effectively gets short sequences of DNA that are unique to particular organisms. You can then work out and identify, if you have even a small part of an organism, what species it came from. So you can use it to look at endangered species. It's obviously crucial to taxonomic research and biodiversity. You can use it to look at agricultural pests. So if you have a single egg, you don't have to muck around. You can find out very quickly exactly what the organism is. And you do that by using this vast collection of information. And so that, I hope, convinces you that this matters. In my heart, it matters very profoundly, simply for the scientific understanding it conveys, but I think it's quite easy to make a coherent argument that it's important in its societal impact. In response to this need and all of this data, an international data infrastructure has evolved around the world, and that's what we've been involved in at the EBI and with other collaborators around the world. And so, for example, for the DNA collections, there's now a very long-standing collaboration. There's a centre in Europe, there's a centre in the USA, and there's a centre in Japan. They're each working on collecting this information, and they share it all on a daily basis. And for the core information, that kind of model is repeated. So for protein sequences, there is a similar tripartite collaboration. It doesn't happen to involve exactly the same partners, but it's basically a long-standing part of the global information infrastructure. The same goes for the Worldwide Protein Data Bank, WWPDB. This is the macromolecular structures. There is, in fact, a three-way collaboration that collects and shares that information around the world. And this global infrastructure is crucial to the science. When I say all this, it sounds a bit as if, well, there's just these databases and they're all kind of the same. It's worth stressing that actually I've talked about a number of kinds of data here, and there's huge differences between them. So you're sharing anything from just a few kilobytes to petabytes of data. If you look at the protein structure database, if it takes 50 new accessions in a week, it's doing quite well. If you look at the DNA sequence database, it's probably taking 5 new accessions a second. So it's rather different. If you look at the costs, I don't know, you might say $5 for a bit of DNA. Determining a protein structure might well be $100,000 worth of science. These are shared in various configurations and collaborations. Some of them are simply centralized at one data center. There are some collaborations that involve 20 centers around the world. Some of them have a handful of specialist users, a few hundred. Some of them have 3 million users. So this is actually quite a diverse situation. So we collect all that information, and we organize it, and we serve it to the scientific community, and that includes the rest of the world and Australia. This infrastructure is a global infrastructure. We tend to serve Europe, but in fact I should say that the second biggest user country of the European Bioinformatics Institute services is the USA. So it is truly global. And I'm now going to be a little bit EBI-centric and also a little bit more try and get to some of the technology of this that enables us to do all this. So we have a series of collections of different kinds of data, perhaps structures, sequences, interactions, or whatever, and users who specialize in them will go through some special interface to get to the data that they care about, and that's historically the way it's been done. In fact, biologists care less and less about looking at things in individual databases, but rather about whole systems, and organizations like the EBI build general search and browse tools so that if you pose a query, you can actually get information from all of the databases that's relevant to that query. And this is part of the way we serve this information at the EBI. And this is happening at the EBI and other centers, and of course for each of these there's probably the kind of global collaboration I was talking about going on in the background. Also, life has become a little bit complicated because actually, historically, the underlying scientific archive was what the users saw, but actually in today's science, say in sequencing, there's all sorts of repetition and projects whose raw data are not of particular use to you, so often there is actually a sort of digest of the information that's built at an organization like the EBI, which is kind of today's best bet about how all the science fits together. So that's the kind of structure you see at the EBI, and relatively recently, there was an interest in involving Australia in that infrastructure, and the first incarnation of this was that, that at the University of Queensland we'd somehow mirror what happened at the EBI to serve Australian science. Now, that has not actually proven to be straightforward to do across the board, and what's happened is that actually some of the stuff at the EBI is mirrored, but users of the mirror will be referred back to the EBI for some of the things that hasn't been mirrored. So it's not a complete functionality at present. However, it's useful because through the mirror, in a sense, EMBL Australia has created a natural connection with the European activities, and the UQ has made the organisational connection with the EBI, and we've done the technical development to demonstrate that we can work together and do these things between Europe and Australia. And in fact, Mark Reagan and a team led by Gavin Graham at UQ are what have made this happen. So there is a sort of complex information infrastructure, and Australia is engaging at it at some level through the connection with EMBL Australia and the technical capabilities there. So what next? Well, before we think about what next, it's worth talking about the scale of this operation and what it involves. If you look at the DNA sequence data, where it's going from a sort of IT perspective, you can say that actually life sciences are going to become a major data-intensive science, easily comparable to say high-energy physics. If we do a couple of 100,000 tumor genomes or something like that, then we're working on towards the exabyte range of data. So this is going to strain our IT solutions. At periods, that data collection has been doubling in size about every 4 months. So, I mean, in IT, people tend to think of the long-quoted Moore's law that every 18 months, what you can get for your money doubles. Well, if you're doubling every 4 months, you're not going to be saved by developments in IT. If you look at the EBI side of this infrastructure, the EBI is actually a fairly significant service center. So it has in its various computer center, it has about 23,000 cores. It's got about 75 terabytes of memory, 19 petabytes of storage, and it's spending about $60 million a year on a 500 staff operation, 3/4 of whom are dedicated to this infrastructural task. The National Center for Biotechnology Information in the USA is of comparable size, and the question arises, where will Australia fit within all this? If you look at usage, just to give you a handle on it, the EBI is serving this information out to a scientific community. Around now, actually I think we're now, at the EBI, they're now seeing typically over 6 million web hits a day, and in 2011 there were about 3 million unique user sites. I actually persuaded them to do a little bit of analysis of the Australian usage for me, and about, from Australia, the EBI's services are currently seeing about 60,000 hits a day. In May 2012, there was about 6,000 unique hosts from Australia that utilised these services. In 2011, there were about 40,000 unique hosts that accessed the EBI services from Australia. So the Australians care about this. So we've got the Australian mirror. It's demonstrated the ability to work together, has a few hundred users, a few thousand hits. What next? It's not just the quantity that makes this task difficult, it's also the complexity. And I gaily drew this picture for you of how these databases are all working wonderfully together. In fact, historically, the way this started was as a kind of parts list, so you'd have a DNA sequence database. People who used it would know how to use it, and if they wanted to use a protein structure database, they'd somehow have to learn the appropriate interface of it, and it was all rather unjoined up. And in fact, our users constantly gave us grief about this, and I like to use this slide because this looks like it's a cartoon. This is actually real. This is actually a genuine map of connections between databases at the EBI. And we were failing our users who were not succeeding in navigating it. And what it amounts to is that this bit, general sort of integrative access to all of this information in a way that enables people to explore it, is the hard bit of the task, and an increasingly important bit, because scientists are far beyond saying, okay, I'm a specialist in DNA. They want to understand entire systems. From an IT perspective, we at the EBI realized that actually we were moving into a new way of working which helped us with this. And for those of you who care, I would call that service-oriented architectures. And what it basically means is that you can build interfaces to databases using standardized technology, and indeed build interfaces between components using that technology and interfaces on the way in using that technology. And the fact that you've got it standardized enables you to modularize things in a way that gives greater capability. The kind of things that I say service-oriented architecture, the way that that's manifested at present is in web services interfaces between these tools, and this creates some opportunities for us. So if you're working that way with that kind of architecture, then when an organization like the EBI is building this kind of structure, it's fairly obvious that it's going to be a damn sight easier for them if they standardize the interfaces to these things and utilize that technology throughout. And so basically, there's an era of IT opportunity, because in IT terms, we know how to do this job an awful lot better than we did 10 years ago. It's worth saying as an aside that when people like me rabbit on about this technology, that the technology won't actually solve the problem without reference to the content. In other words, I can have all sorts of clever interoperability technology, but if you don't have good ways of working out what in this database belongs with this object in this database, then you'll never make the connection. And there are a number of projects and issues you have to deal with to enable that. And perhaps one of the most fashionable fads is for the sort of ontological activities that try and standardize terminology in a particular area. There are other aspects and other things that you can do to enable that integration, but this one is an interesting source of tension in the biological world, because actually there is a whole sort of formalism around ontological work, and we require a kind of pragmatic solution that works well, and often there's a bit of tension about people going away to try and do ontologies and then discovering that they've involved themselves in trying to map out the entire knowledge of biology in an ontology, and they never quite come back from the task. So these are that's one side of technical changes in the IT world, which I think enables us to do this job better and differently in the future. There is another change in the IT world that's important to this, which in a way you could almost say the EBI adopted by accident. In a scaling up exercise, the EBI is serving a large number of users. It had a fairly significant data center on its site, but was outgrowing that and decided it needed to move off and take advantage of facilities elsewhere. So we were doing a major scale up, and it turned out that one of the things that had developed within the IT world that made that easy was the concept of virtualization. So what this means, and the IT people will criticize me for my simplification, is that it would be nice if all computers were the same, because then any program could run on any computer. That's not going to happen, but there is a way around that. You can say, actually, here's a definition of a machine, which is going to be the standard machine. And even if you're not one of those guys, you've got to be prepared to pretend to be one of those guys. And that means that you can, if you've gone into this concept of virtual machines, you can do your computing where the hell you like, and you quickly get to the sort of view of the world that, well, actually, do we even need to own any computers? Now this, from an IT infrastructure perspective, in a sense, if you talk to the e-science people, they would be rather excited by it. Because if you've got what I referred to the service-oriented architectures and virtualization, to my mind, you've almost invented cloud computing, which is one of the great IT developments of today. And so you could argue that that's the way forward to solve this problem in collaboration with your IT guys. And in fact, we already have the Ensemble database in the Amazon cloud, and actually the USA group have put the 1,000 genome database in the Amazon cloud. This is actually, the Amazon cloud is not a cost-effective solution unless they're prepared, unless they want you badly enough to do it on the cheap. Now, an interesting thing about this emerging technology from an Australian or perhaps anyone's perspective is that we've now, through our service-oriented architectures, effectively evolved a standardized way of exposing our databases on the network. And we're doing that in order to cope with our internal engineering, but that creates an opportunity, because if that interface is public, then okay, the EBI is building its clever tools to access it, but in fact, there's no real reason why somebody else can't use the interface and say, actually, we'd like to build it. We think we could do a good job of building tools to access the information. And so that creates a new kind of opportunity. This is also an opportunity within Europe and pan-European infrastructure. And for those of you who get involved in the global discussion of how to do these tasks, you may have heard of a project called Elixir, which is a big collaborative project which the European Union is funding. And basically, this is based on a realization that the scale and complexity of the information infrastructure we require overwhelms the capability of any one organization, even organizations as big as the EBI and NCBI. And so in Europe, new structures are kind of emerging to share this infrastructural task under this title, Elixir. Now, the notion with the Elixir is, and these slides are stolen from the project leader, Dame Janet Thorne, these are, is that coordinated funding at centres throughout Europe with a hub at the EBI can share the task in an effective way. And the UK at least has put their money where their mouth is, and that they've just given us 75 million to build the hub at the EBI, or given, I keep saying us because I used to work at the EBI, I mean them. So 57 different nodes throughout the 24 different countries have expressed interest in being involved in this, either because they have specialist information that they think they can feed in, or because they want to serve a local community or whatever. There's different motivations for them. And actually, 11 of them have signed up, 11 of these countries have signed up in a way that says they'll put some money into it. There's this global infrastructure, there's machinations in Europe about how that's going to go forward, and we want to share this task in a way that makes it effective. And really this, what it amounts to, is we'd like to get our hands on all the resources we can to do this task together. And if there's a motivation for Australia to be involved and contribute to the task, that would excite us. And I hope that we can use Australian resources effectively. So I've been involved in this discussion, and I am likely to come and work for a while in Australia to work out how Australia might engage with this and what the motivations for Australian engagement would be. And I have, at this stage, all I have in my head is possibilities rather than strong opinions, or at least I have some strong opinions, but they change frequently, though not the fact that I'm right. And so basically, there are questions about this task and the motivation for Australian involvement. And at the simplest level, if Australia can do some of the work, then that ought to be a good thing. Another motivation is simply to optimize Australian access to this information. If I need to do the investigation to see whether this is a real concern, but we certainly want to be sure that Australian scientists are in no way disadvantaged in their ability to exploit all this information. If indeed this information is visible to the world, then obviously Australian science ought to be visible in those databases. But I would like to think that it goes further than that there's ingenuity in Australia that basically could say, okay, we can do some clever things to enable new ways of exploiting the information. And there are certainly some people who feel, and you can imagine that if Australia is contributing to this global endeavour, then they get to be involved in how it develops over time. And these are all questions for Australia, and the answer to some of them would say, well, actually, why would we have that ambition? But if we look at sharing the task, and I've described, let's think about ways that Australia might be involved in sharing the task. I talked about these global collaborations, and you could say, well, okay, if there's a DNA sequence or a protein sequence collaboration, should Australia actually be bidding to be a member of one of these collaborations? My initial view of that would be that it's actually fairly unlikely to make sense. If there's a structure that's working in the globe, then what benefit would be there to be had by Australia being involved? If there are new kinds of information that come along, I don't know, I gather from the scientists that there may be in the sort of RNA world, then it may make sense for Australia to say, actually, we could contribute in that new collaboration, so that there may be possibilities for sharing the task. In optimizing Australian access, well, we have actually done some mirroring work. We can also do things to do with training, with user support, and if there are indeed delays in networks and bottlenecks and things like that, we could work on those. And if I were to ask to express opinions about this, I would say that the notion of a mirror for the sake of a mirror, of the kind of activities that the EBI is involved in, makes sense if that's the best way to get the performance you need. It should be used if it's the tool that's required. It's fairly obvious that it's a good thing to be involved in training people how to use these resources. And it's fairly obvious that if there are network and IT bottlenecks, then some of the resource that's being spent on e-science in Australia ought to regard it as part of its responsibility, in my opinion, to solve these problems for the life sciences. There has been some of the people I've already spoken to has sort of said, well, we need our Australian scientific results to be visible in the databases. I find that interesting. In truth, the databases are already global, and anything that Australians do that should be in the existing public databases indeed already is in them, so it seems a wee bit of a non-question in some ways. You could argue that data of particular Australian interest should be contributed and curated by Australia, and I think that that's just fine, and probably the global structure would expect that, but I don't think that Australian ambition should be constrained by this. I think Australia is a grown-up scientific nation that should be working with all the things that everyone else is working with. Australian ingenuity, well, I did say that the way the IT is evolving, it's far less of a closed shop how you build tools to access the data. And if you were to ask me about whether that's a good thing, I would say it's perfectly fine for people in Australia to put some energy and dedicated R&D work into developing tools, because there's no one right way to do this. And in fact, one of the things that I think is important is I feel that in the underlying data infrastructure, it's fairly clear that you want to have a unified single instance of that, if you like, but actually in tool building, you can argue that a bit of competition is a perfectly fine thing. If you think that you can do something clever, why not build the tool and let the market decide? So that's something that we could consider as a worthwhile Australian activity. In terms of the sort of global setup that we have in Europe, is it interesting to Australia to be involved in that? That's a question that needs to be answered. I can imagine two different levels of involvement in it. One would be to say, okay, let's talk to these Elixir people and say that there should be non-European and Australia could be a spoke of the Elixir operation, or if you were being even more ambitious, you could say, well, there ought to be a hub for that, an equivalent hub in the Southern Hemisphere. That's an open question as to whether to do that. These are all my thoughts about this task. I talk about infrastructure, information infrastructure. Infrastructure tends to be a word that has rather specific meaning to funders. Really what I'm talking about is the kit, the people, the engineering to do this task. Part of my responsibility in the coming year or so will be to work out what, if any, the correct Australian engagement with that is. This is all fairly abstract stuff, and I would like to just sort of conclude by saying that actually we can draw pretty pictures and nice arrows and all this kind of stuff as long as we like, but there's really very little point in doing anything unless Australian, we'd be pretty pleased if Australian bioinformaticians were actually saying that this infrastructural resource we built in Australia is useful. Actually, I personally would be 10 times as pleased if Australian biologists, not Australian bioinformaticians, were saying that it was useful. And in fact, just before I left the EBI, we did the last ever user survey that we did. I was actually quite delighted that for the first time ever, more than half of the people using those resources didn't describe themselves as bioinformaticians. They said they were biologists, and that pleased me immensely. It would be nice, if we're going to do something useful in this context, that actually the biologists worldwide really sort of said the place to go for this particular thing is Australia, because they're best in the world at it. This is, as I say, an infrastructural task. Australian ambition is yet to be defined. If I'm going to be involved in it, I'd like it to have a big ambition. But let's take it from there. Thank you.
Thank you, Graham.
Thank you.
And I mentioned at an earlier event that the Life Science Forum was putting together a publication which you may have picked up a copy of out the front. So this is really an opportunity to share with the community why Melbourne is such a great place for the new biology, the convergence of biology, and some of the things that we've heard from Graham this evening. I want to thank the sponsors of the Life Science Forum who made this publication possible, and also Neil Byrne and his team at Science and Public, who really put together what I think is a very good read and a compelling story of why Melbourne is such a great place for this research. And as usual, to thank the sponsors of the forum who make this event possible. So again, thank you for your company and support this evening. And again, please join with me in thanking Graham for his presentation. Thank you very much, Graham.